Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. My name is Ahmad Syabian Zahar, meter number 202316671, and I will present about the practical one. The objective of this practical are to calculate and describe descriptive statistic, mean, variance, and standard deviation, and to understand the concept of correlation and determine the relationship between the two variables, leaf length and width. The material we use are leaves, ruler, weighing, balance, graph paper. The method involves measuring the length and width of 10 leaves in 5 sets, calculating mean variance and standard deviation for each set. A data matrix is created with this statistic. A scatter plot is then constructed using all the leaf measurements to analyze the relationship between length and width. Then, I will describe the outcome of practical 1 using 3 sets of leaves. To begin, the first mean result is determined by summing all the leaf length divided by the number of leaves. And the correct answer is 23.2. Variance may be calculated by subtracting the result of the leaf length addition from the mean. After that, multiply by 2 and divide by 9. 3.733 is the answer. The standard deviation may then be calculated by taking the square root of 2 variance, which is 1.9321. We go to the leaf width table in the same manner, and the mean and variance of the leaf width are 6.6 and 7.7922. Respectively, 2.8234 is the standard deviation for table leaf width. For sets 2 and 3, this technique of computation will be used to determine the width and length of the leaf table. In table 2, the mean and variance value for leaf length are 21.1 and 6.911, whereas the standard deviation value for leaf length in set 2 is 2.62. As a result, the mean and variation of the leaf width of set 2 are 5 and 1.111, respectively. And this is the outcome for set 3 leaf length and width. This is the graph of leaf length versus leaf width. There is no relationship between these two variables.
Assalamualaikum warahmatullahi wabarakatuh. My name is Anis Rabinti Karim-Anwar and my student ID number is 023168461. So today I would like to present about the first question for practical 1. The first question is, describe the differences or similarity of the mean of length between different sets of leaf. Do the same for the mean of width. On the graph, there were differences in means of the length between 3 sets of leaf. The mean of length of leaf were 23.2, 21.1, and 22.4 for set 1, set 2, and set 3, respectively. These differences may be due to the growth stage. This is because the mean length of leaf can vary depending on the growth stage of the plant. The mean width of leaf were 6.6 for set 1, 5 for set 2, and 5.85 for set 3. This may be due to environmental factors. The second question is, what is meant by variance? The leaf have different measurement of width and length. What is the meaning of this statement? Discuss with respect to variance what is meant by standard deviation. According to this experiment, there are 10 leaves for each 3 sets, which will have 30 leaves in total. The total value for width and length is 66 cm and 232 cm, 50 cm and 211 cm, 58.5 cm and 22.4 cm for set 1, set 2, and set 3, respectively. Furthermore, the value of variance in length and width for each set is set 1 is the 3.733 and 7.972. Set 2 is 6.911 and 1.1.1, and set 3 is 11.1 and 0.8361. The standard deviation is defined as a measure of amounts of the variation or dispersion of a set of values. The third question is, why is it necessary to have different set of measurement? For the measurement of leaf width and length, it is necessary to have different sets of measurement for the measurement of leaf width and length to get a result that shows whether the plant contains the same characteristic or not. It is also necessary in order to obtain valid data and results where we can have an average measurement of leaf instead of only using few leaf. So that is all from me. Thank you.
Here are some explanations about the practical 2, which is factors that affect the distributions and abundance of organisms. The objective of this practical are to introduce the simple concept of quadrats, enhance the power of observation, record and note the difference in plant distributions within the quadrats, and to determine from natural observations of the potential factors that influence on the quantity and distributions of the organisms. Thence, the materials that we are used to complete the activity of practical 2, which are referees, measurement tape, stick, scissor, and notebook.
First step in the method process is the different types of forest and eco-rainbow wool bundle will be the area under study. Next, make 1 quadrat of 5 meter times 5 meter at each forest type. Multiple quadrat samples will be performed throughout the habitat at several random locations. Third, observe and take note of all tree stands that are found inside the quadrat, which must select areas that have a wide variety of plants. After that, measure the diameter of grass height for each of the trees that stand within your quadrat. Lastly, data will be used to estimate the population size and population density within the entire habitat. In the result quadrant, we found 8 different species of plants. 4 species such as Diplezium and Idea. The total number of individuals is 4, and this species disperse a wide range. The density is 0.16. For species such as Setaria palmifolia, Orchanostasis, Amantaceae, and Bamboo, the total number of individuals is 1, and these species do not disperse. The density is 0.04. For species such as Disporos, Buphsofolia, and Bennett's ash, the total number of individuals is 3, and these species also do not disperse. The density is 0.12. Lastly, for species Bissa silanica, the total number of individuals is 2, and the density is 0.08. A quadrant is a firm traditional square used in ecology, geography, and biology to isolate a standard unit of area for the study of the distribution of an item over a larger area.
For the second questions, it asks us what are the 2 basic requirements of all the quadrant techniques. The 2 basic requirements of all the quadrant techniques are the areas of volume counted in nonce so that density is determined directly, and the second one is the organisms are relatively immobile during the counting period, so none are missed. Moreover, question 3 say what would be the advantages and disadvantages of using a larger or smaller quadrats to survey the sedentary organism. For smaller quadrats, the advantages is the sampling is easy to use and inexpensive. It refers to the quadrat sampling application with advantages and disadvantages in 2020. For the disadvantages of smaller quadrat, it is might miss too many individuals, resulting in other representative estimates of the populations. Furthermore, for the larger quadrats, the advantage that we can find is larger quadrats cover a larger area, it allowing for the inclusions of more individuals or species in the sample. Next, the disadvantages of the larger quadrats is larger quadrats can encompass more heterogeneity within the sample area, resulting in increased variability in the data. We dive to questions 4 that asks us to explain the issues concerns ecologists in conducting sampling method. The first issue is sample representativeness. Ecologists aim to gather data that accurately reflects the entire ecosystem or populations being studied. However, achieving the representative samples can be challenging due to the inherent variability within ecosystem. Therefore, the second issue is sampling bias. Sampling bias occurs when certain individuals or groups within a population are systematically over or underrepresented in the sample. This can skew the results and lead to the inaccurate interpretations.
There are several factors that can contribute to this variation. First is habitat diversity, where the diversity of plant species in a quadrant can be influenced by the range of habitats present within the area. Second is disturbance and succession, which can influence plant species diversity. In areas with recent disturbances, few plant species may be present as the ecosystem undergoes recovery. Next is geographic factors, where the geographic location of a quadrant plays a role in determining the available pool of plant species. Lastly is the sampling effect, which the number of plant species recorded in a quadrant can also be influenced by the sampling effect, including the size of quadrant, the number of predicates, and the trueness of the survey.